Okay. All right, so this is a Kahoot on this one. So if you guys have your, whatever phones or computers or whatever, we're going to do that. It's like slides and Kahoot at the same time. It's so violent. In the road between, I couldn't drive my SUV. I'm addicted, so I couldn't get my husband to drive, and I couldn't find the key. I'm not gonna use cows on the SUV. Really like the Tuesday class, which is on 81 there. Me trying to get over my chest anxiety, really halting. So. I live in Arcadia. I'm second. Okay, now. All right. Is that everyone that's gonna join? No, I haven't yet. Okay. I'm not in any hurry. You're fine. All right, we ready? Okay, super fun.
Ready? Keep in mind, you guys can join during the game too. You just have to use the number. You won't have to think. All right, so this is going to be kind of divided down into sections, essentially like the safety section, and there will be like, I don't know what is, there's a DNA section and whatever. So, and then the ELISA section. So starting off, we're going to do the safety section, basically the safety rules that you're expected to know. You could be quizzed on those on the test. So for example, where should broken glass be disposed? Right, so that's going to be in the sharps container. All right, moving on. That's it for that. We're going to review for the safety. Just be sure you brush up on the safety steps. We can't run and go over everything. But getting to the stains, what you guys should know about the stains. You want to know the steps in order for each of the stains. You don't need to know the times, but yeah, be aware of the steps and kind of what the purpose of the step is for the stain. And then positive and negative stains, what that means and what they do. And what happens if you skip a step in any of these? So like if you're doing the gram stain, what would happen if you skip the iodine step? What happens if you skip the alcohol, which is a more common question that would happen, but yeah. All right, so let's go through. What is the purpose of heat fixing? Select all that apply. All right, so the answer is going to be adhere to the slide, which we kind of know about this, we can stick on there really well, as well as kill it. So we kill the actual bacteria, so it's not going to be, infectious or something. It also prepares it for staining, but it doesn't stain it. What is the counter stain that we use in gram stain? Yep, so safranin is the counter stain. So basically, like the main stain is that crystal violet. We do the alcohol rinse because gram negative, it'll rinse that out. So we counter stain with the safranin to see what's going on with that. So I'm probably going to ask a question about that further. Yeah, what color will a gram negative bacterium be after the gram stain? All right, so now you basically had two choices here, right? It's either red or purple. So the gram negatives are going to be red. The positives will be purple. All right, what is the first stain used in the capsule stain? All right, so once you guys got that one, that is correct. It's going to be Congo red, and it is used for a very specific reason, which is the next question. I always jump ahead and like start talking more about it, and then it's the next question. All right, what type of stain is Congo red? So you'll select all that apply to it. All right, so right, so it's gonna be background, and then you guys did really good with that, actually. A lot of people get it mixed up, positive or the negative, but you guys did really good with that. Remember negative stain, the cell, outside the cell is negative, right? We've got the phosphates on there with the phospholipid. So they are going to repel negatively charged stains, and that is really what these are about, whether they're negatively charged or positively charged. So you're staining either the cell with the positively charged or the negatively charged stains with background. Good job, Ben. All right. The following is a pure culture. What stain is pictured? All right, good job, guys. That is the endospore stain. Pretty obvious endospores in there, I feel like. Good stain. Right, there could be more than one answer for this one. What is the term for the red structures in that same image? The red part. All right, yeah, most guys got that one. That's correct. This is not supposed to be marked. I'm sorry, that was a mistake on my part. The red structures are going to be the vegetative cells. That's in the green. And that's going to be that counter stain there that's being used is the safranin again. All right, which of the following bacteria would be acid fast positive? All right, the answer for this one is Mycobacterium flea. That would be, you know, somewhat related, I guess you could say, to Mycobacterium tuberculosis or leprae. So, yeah, it's gonna be Mycobacterium always for that one. All right, what is the primary stain used in the acid fast stain? Yeah, so this one is going to be carbofusion. I mean, that's just a memory thing. So remember that carbofusion is going to have the ability to interact with the mycolic acid, which is that waxy. In which staining procedure did we use C? So that is going to be the endospore stain. Which of the following structures allow for bacterial motility? For bacteria. Good job. That is going to be the flagella and only the flagella. You can have more than one. And they have those nifty arrangements with all those fancy words. Okay, with the stain, here's the stains that you need to be familiar with. The simple stain, the gram stain, the acid fast stain, the capsule stain, such as, if we're talking about capsule stain, why do we not fix it? You might want to order stuff like that, okay? Wet mounts and then endospores stains. And why we do all the crap associated with it. All right, next we're going to move on to ELISA, which we just did, because this should be, I'd say it should be pretty easy, but people still have a hard time with it. What does the E stand for in ELISA? So yeah, it's going to be enzyme linked immunosorbent assay. What is an indirect ELISA looking for? What is it testing? It's the one we did in the simulation. All right, so this is going to be testing whether or not the patient has the antibody. Good job. You guys did better than the last class. All right. With ELISA, it's not just going to be about ELISA, right? There's other like tests that we learned about, like rapid testing around that same time. But yeah, you want to be familiar with the general concept of what antibodies and antigens do and how they interact. So, you know, so you get ELISA and how it works. Indirect versus direct, which you guys got pretty good. And then general theory on how the assay works and color change and all that. Okay. So you do have to know some DNA stuff. There's the stuff that we tested about online, like PCR. We did not do in class, but we did do that in the online, whatever, homework, which we learned also in the lecture, just to be fair. And then transformation simulation, you'd be aware of that one too. So what does PCR do? Good job. So strange looking at it from like class to class. My last class was like all in on the blue. So it's pretty interesting how it gets spread out. So yes, polymerase chain reaction is what that stands for. Polymerase is referring to DNA polymerase. So we're just amplifying DNA. Oh my gosh, here we go. Sorry, the mouse is hard to operate. Which DNA fragment is the largest? All right, good job. That is correct. It's going to be 35 kilobase pairs, 35,000 base pairs. I don't know what it was like the last couple of semesters. Nobody was getting the concept of this, like the kilobase pairs versus the regular. But I'm glad that you guys are not struggling with that, because I did not understand that, why people were struggling. All right, which of the following DNA fragments would travel the farthest on an agarose gel? Yeah, okay. All right, what type of cells are capable of taking up exogenous DNA? So that's going to be, they're going to be called competent cells. So that just means they're able to take up DNA from outside around them. And they can be any kind of So. All right, as far as the DNA stuff goes, the DNA structure and function, I really don't need you to focus on that too much for the lab, okay? It's more of like a collection thing. DNA fragment sizes, which we just talked about, as well as the gel electrophoresis principles. I would also know like why DNA moves to the direction that it does in that. PCR works in a practical sense. Oh yeah, so like the steps. So like the three main steps of PCR are going to be like when you heat it, high heat will denature it, that will separate the strands. Then they say annealing is the next step. That's when we sticking some primers on because DNA polymerase doesn't just work without help. There has to be something there to build off of. And then elongation, which is pretty straightforward. So that's basic steps, and you can just keep doing that over and over again in PCR. And then, oh, another thing I would say about that. So if we had one DNA molecule, right, and we wanted to PCR one round, then we would have two at the end of the round. Yes. So I would be aware of that for the test. I'm just saying it will ask you at what round we will have how many. Okay. Forgot about that for the last test, for the last class. But you guys get the heads up. Eukaryotes. So here you're going to review your drawings. You know, remember the pictures of what you saw in the actual microscopes for this, but we're going to go over a few of them here. So which organism is this? This is pretty much what you're going to see for that stuff on the test.
Are they going to be as easy as this?
I mean, some of them will be. Yeah.
Okay.
All right, so that is correct. Once you guys got that one, again, Aspergillus, good guess. They both are one of the moles, like the fungus, right? You guys did good. That's penicillin. Just be sure you can tell the difference between those two. What is the name of this organism? Yeah, so that is exactly right. That is Giardia. So it looks sort of like a horseshoe crab, and they're pretty small in actual slides. So if they do come up as part of a question, just be aware that they might be small. All right. Remember the organisms you had to draw. Important structural and reproductive differences that might have been mentioned in the little worksheet. Trophozoites versus cysts. I don't need you to know, like, I need you to know trophozoites, what they look like, but the cysts, like the concept of the difference between the two, right? And then the identity based on the differences between them. There's like a talk about like life cycle on a couple of them. Review that stuff, the written stuff, the stuff that wasn't in the actual like microscope. Okay, general bacterial growth. So we're going to get into general media use and culturing. Which of the following is not a term used to describe colony morphology? All right, so this is going to be hyphae. That's referring to the branching off that we get in mold, like the actual structure of it, not like colony morphology of it. Colony morphology is talking about what the stuff looks like, on face value. So be aware of these terms. We're going to get into the other terms here in a moment with like cell shape and arrangement. It's like there's one station I know that I have like a question about this colony morphology, a question about a cell shape, and a question about cell arrangement. I'm like pretty sure that they're all like kind of in a row. What is the name of this type of culture? And keep in mind that like on the test you'll have actual things watch your hands up. So it'll be easier to know. But yeah, that's a slant in that. How was this organism isolated? Good to know. Replay. Be aware of which quadrant would have the most single colonies. So which arrangement of bacteria do you observe? With what kind of arrangement? All right, so this one's going to be strepto. It's the chain. What is the name of the shape of these bacteria? This is spirillum, but that little kind of S shape, sort of spiral shape. But on the exam, you'll only see either cocci or bacilli. I'll just be honest. I don't have any other kinds. Okay, so with that general bacterial growth stuff, colony morphology, just be familiar with the terms. Like this is what terms are part of referring to colony morphology. The different kinds of media, and there we might be talking about the general idea of selective and differential. I know we're going to get to the details of that after the exam, but general details. Good. Isolation technique, cell shapes. Remember with isolation techniques, it's not only going to be the street plate. Okay, there's other ways to isolate things like the serial dilution. All right. Cell shapes and arrangements, just be sure you don't get them confused. And in microscopes and how to use them properly. If you observe a specimen at 40x objective and have a 10x ocular lens, what is the total magnification of that specimen? So that is going to be 400x. You just multiply for this one. Where's my cursor? There it is. Which objective lens do we use for oil immersion? Right. Well, yeah, we all know that one, right? If you didn't know that one, I'd be a little worried for you guys. Okay, be able to calculate the magnification, which we just did. Understand the operation of the microscopes, know all of the parts and how to use the parts with the correct terms for them. There will be a microscope and there will be at least one label on it asking you to identify the parts, okay, and what it does. And then understand why we use oil immersion, just to prevent reduce light scatter. There was something else I wanted to say. What is this number? All right, so on to Kirby-Bauer. This is our antibiotic susceptibility. Which antibiotic is this bacterium the least susceptible to? All right, that is correct. That's going to be E. Because again, clearly it wasn't affected by it at all, right? That's the idea there. All right, with Kirby-Bauer stuff, be aware of what ZOI means. That's zone of inhibition. That's the area where there was no growth around the disc. That's what we measured. And then we have the minimum inhibitory concentration. We can calculate the minimum amount of antibiotic it takes to inhibit that bacterium for each bacterium using the ZOI measure. We didn't do that. It's just a thing that we could do. Mueller-Hinton auger, that it's all going to be for the same level and the same thickness and with pH controlled so that the antibiotics can spread evenly. And then bactericidal versus bacteriostatic, know the difference. And if I were to say, if you were to sample around this here, K30, if you were to sample around here, streak it on a new plate, and then it grew, what kind of antibiotic is that? You would say bacteriostatic, right? You would know that over a cycle. Just be able to apply that. All right. So that is that for the review for the lab. I mean, like I said, it's going to take 30 minutes to even took that. But yeah, do you guys have any questions about that? It'll be on Scantron. I have Scantron sheets I will provide for you. You do not need to buy your own Scantron sheets. Do bring your own number 2 pencils though. I have some, so if you forget, I can supply, it's okay. But just be sure, like yours will have better eraser, is what I'm trying to say. You know how the ones that will just smear it around with all of these. So yeah, I recommend bringing your own pencil if you want that eraser to work. And it'll be regular lab time. So instead of lab, it'll be the test. Okay. So that'll be on Wednesday. I can't believe it, but yeah, it'll be Wednesday. All right, here is our little podium in case anybody cares over it. Here. Where is my paper? What's been up there? This is like a game in and of itself, finding my cursor. When it's over there. All right. All right, let's stop. Yeah, so that's it for the review for the lab. If you guys are staying to take the test, you are either welcome to study in here and until you want to. I mean, I don't know. Let me take a vote actually.